Hi, my name is Aoi Sasaki. Today I'm presenting a review of the paper titled The Gut-Brain Axis-Mediated Sugar Preference. Sugar is a fundamental source of energy and motivates us its consumption. Most species have evolved dedicated brain circuit to seek sugar. Surprisingly, even in the absence of a functional sweet taste pathway, animals can still acquire a preference for sugar. Although artificial sweeteners activate the same sweet taste receptor as sugars, they fail to substitute for sugar in generating a behavior preference. From those results, the authors hypothesized the existence of a sugar-specific rather than sweet taste-specific pathway that operate independently of the sense of taste to create preference for sugar. In the first experiment, they tested sugar preference by using taste knockout mice. They led mice to drink from two bottles which contain either sugar or sweetener. As the graph shows, even though taste knockout mice cannot taste sugar and sweetener, they learn to recognize and choose to drink sugar solution more than sweetener solution. In the next experiment, they tested the population of neurons that respond selectively to the consumption of sugar. They exposed several cohorts of mice to sugar, sweetener, or water and examined their neuronal activities. They tested the brain region called caudal nucleus of solitary tract (CNSD), which is known to function as a nexus of interoceptive signals conveying information from the body to the brain. The results showed that significantly higher number of neuronal activities in CNSD after mice had sugar compared to sweetener. So the next question was, how do sugar signals reach to the CNSD in the brain? From the previous experiments, they assumed that intragastric infusion of glucose is sufficient to condition flavor preference. A number of recent studies have implicated the gut-brain axis as a key mechanism for transmitting information from the gut to the brain through the vagus nerve. In this experiment, sugar or sweetener was infused directly into the mice's gut and measured the activity of CNSD depending on with or without intact vagus nerve. As they predicted, the results showed robust responses to sugar and sugar analog. Remarkably, all the activity was abolished after bilateral transection of the vagus nerve. They also measured the sugar preference when they were silencing the neuron in the CNSD. Optogenetic was used to control the activity of neurons. The results showed that silencing the sugar-activated CNSD neurons abolished their preference for sugar over artificial sweetener. These results illustrate the essential role of the circuit in driving the behavior preference for sugar. In the next experiment, the activity of real-time vagal neuron responses to the gut stimulation with sugar was directly monitored by the calcium imaging. Around 200 neurons that display statistically significant response to glucose, but less than 1% of these neurons display stimulus-dependent activity to sweetener. From the results, the authors reasoned that the gut-to-brain signal might depend on non-sugar sensors recruited into this role. So they tested whether pharmacological inhibition of SGLT1 abolished the glucose-dependent neuronal responses. SGLT1 is responsible for glucose absorption in the small intestine, and it is thought to function as a pipe between the gut and the vagus nerve. Fluorescein 19 was used as a blocker of SGLT1. The result demonstrated significant reduction of glucose responses following intestinal application of fluorescein. This result placed SGLT1 as an important component of the sugar preference signaling circuit. Next, they tested whether the selective activation of this circuit can create a preference to a previously less preferred stimulus. First, they identify a genetic driver that marks sugar preference neurons in the CNSD and then linked their activation to the ingestion of novel stimulus. PENK-expressing neuron, which is shown in the red color on the images, responded strongly to the sugar stimulus. So they made PENK CNSD neurons able to be chemogenetically activated by the clozapine. Under this paradigm, the cherry flavor solution was made sweeter than grape solution, so that the animals are more attracted to the cherry flavor. Next, they introduced clozapine into less preferred grape flavor and investigated whether clozapine immediate activation of the PENK CNSD neuron can create a new preference. After 48 hours of exposure to the grape and clozapine bottle, mice completely switched their preference even though the grape solution was far less sweet than the cherry solution. These results demonstrated that artificial activation of the sugar preference circuit is sufficient to drive the development of the previously low preferred stimulus. In this study, authors demonstrated that the glucose acts in the gut to activate the neuronal circuit that communicates to the brain the presence of sugar. A post-ingestive system in the gut assures that signaling only occurs after the sugar molecule reaches their desired target for effective absorption and metabolic consumption. This is the information of the paper that I presented today. Thank you.